It is the most commonly used recreational drug in New Zealand. Yes, it's a drug, and many people don't understand that alcohol can ruin your life in just as many ways as drugs like cannabis, perhaps even more. Ooh. 1 in 4 Kiwis are considered to be heavy drinkers, and 250,000 New Zealanders are chronic alcoholics. And many do not fully understand what alcohol can do to your body and also to society. Our healthcare spending on alcohol-related issues is $4.9 billion per year. Imagine just how much an impact just a little bit of that money could have if spent elsewhere. Alcohol in the brain acts as a neural inhibitor. It reduces the transmission of signals from neuron to neuron. Think of your brain like the post system, letters being received and delivered. Alcohol acts as an interruption to this postal service, so the letter may not arrive on time, to the correct address, or the message may arrive jumbled up. With long-term alcohol use, chemicals and messages in your brain change, and the levels of these messengers change as well. Most significantly, the levels of microRNA in your brain change. MicroRNA, or miRNA, are one type of DNA, or RNA, a copied strand of DNA that is found outside the nucleus of a cell. Certain types of RNAs do different jobs. MicroRNA's job is to prevent the translation of RNA at the ribosomes in a cell. RNAs are read by the ribosomes, and then the ribosomes use the RNA as a template to make enzymes and proteins for the cell. miRNA binds to the RNA traveling to the ribosome, preventing it from being read. It does this in 2 ways. It physically blocks the ribosome from being able to read the RNA, and it also breaks down the poly-A tail of the RNA. All RNA have a poly-A tail, and is used by enzymes to know when to break down the RNA to make new RNAs. It's like a ribosome bus ticket. It gets clipped after being read to a ribosome, and when all the rides are used up, it gets destroyed. The presence of the miRNA breaks down the poly-A tail quicker than it normally would, thus breaking down the RNA quicker, a process called dead annihilation. We know the levels of Mi206, a microRNA, change and increase when alcohol is present for long periods of time. Mi206 stops the translation of brain-derived neurotrophic factor, or BDNF. BDNF helps support the translation, the growth of new neurons, and enables existing neurons to form new connections with other existing neurons. This process is integral to learning and developing, and is called plasticity. Due to the reduction of BDNF, neural pathways degrade. Over time, this can alter a person's short-term memory, personality, decision-making, creating an ingrained neural pathway of alcohol abuse. This is alcoholism. So, how can we reduce the effects of Mi206? Fortunately, yes, there is a gene that inhibits it, called myostatin, and is already coded into our genome. Myostatin breaks down the Mi206 and therefore increases the levels of BDNF in your brain, preserving your neural pathways. But since myostatin is actually used to inhibit muscle growth, artificially increasing myostatin production in your body will not work as a solution. Too much myostatin will break down your muscle, causing muscle atrophy. The myostatin will need to be produced outside the body and applied directly to the affected neurons. We can locate the gene that codes for myostatin, and we can use this to genetically engineer bacteria to produce the myostatin for us. Enzymes cut the bacteria's plasmids and insert the gene for the myostatin gene into these plasmids. The bacteria then divide, producing identical bacteria that make the myostatin. This technology has already been proven to be incredibly successful and reliable, with E. coli bacteria currently being used to make insulin for diabetics. As for delivering the myostatin to the brain, recent advances in nanotechnology have created particles that actively seek out specific tissue or even individual cells. This can be used to deliver the myostatin to the affected areas of the brain without any of the muscle degenerative side effects. So, we've solved long-term alcoholism. Now what? Just by treating 1% of the alcoholics in New Zealand could redirect $49 million per annum back into our economy. This can be used to treat a plethora of issues, such as air or groundwater contamination. Other great economic benefits will be felt in the workplace. Hangovers and alcoholism force many people to spend lots of time off work. Reducing the health impacts of alcoholism will ensure that more people will spend more time in the workforce, increasing workplace productivity and efficiency. Alcohol-related issues will be less of a social detriment. There will be less child neglect. There will be less women as the target of domestic violence. There will be less depression and anxiety, leading to loved ones being lost. With the reduction in the effects of alcoholism this technology promises, New Zealanders can step into a safer and more secure future. Tangible benefits will be felt across New Zealand. Now only one question remains. What are we waiting for?